I will just narrate it, and you can move that forward. You can pull up a chair. There's an extra chair out there, and we can just go. So, okay, so, all right, perfect. All right. All right, so fatty acids are very highly caloric, about, as I mentioned, about 6 times the calories per gram versus glycogen. Okay, can we go to the next slide? They don't require water. I mean, so glycogen, sugars, proteins to some degree all have issues with needing water to surround them, but fats do not. So they're anhydrous. When we talk about fats, we're really talking about triacylglycerols. And so what we have is a central glycerol molecule that has been esterified with 3 fatty acids. And some of those fatty acids, well, some of the fatty acids can be short chain, long chain, fully saturated, or they can be unsaturated or have 1 or more double bonds. Okay, let's go on. Okay, we eat a lot of fat in our diet. The Western diet has become quite caloric. Recommendations are less than 30% of our calories come from fat. My guess is that in many cases, more than 50% of our calories come from fat. A lot of our foods have been enriched with fats so that we enjoy them. They taste good, but they are highly caloric, and that can be a problem in terms of weight. Okay, moving on. All right, we're going to talk about the digestion of triglycerides, fats, in the diet. So they enter the stomach, and actually there are, I think it's recently been found that there are actually a group of enzymes that act in the stomach to break down triglycerides. These triglycerides have short-chain fatty acids or maybe mid-chain fatty acids. These are gastric lipases, but the majority of the digestion takes place in the small intestine. So in the stomach, maybe 10 to 15%, maybe, and in the small intestine, 80 to 90% of the digestion of triglycerides takes place. As the contents of the stomach enter the small intestine, there are a group of molecules called bile salts. We saw these when we talked about lipids, but these have a sterile backbone that has been modified with an acid functional group, typically on what's termed the side chain, this hydrocarbon piece that is an extension of the D ring, the upper right ring. These bile salts act as detergents, emulsifiers. They're able to, the 4 fused ring system of the sterile backbone is hydrophobic, and it can interact favorably with the hydrophobic portion of triglycerides. And the acid moiety, be it a sulfonic acid or a carboxylic acid that's on the bile salt, interacts favorably then with water. So you have the hydrophobic pieces of these bile salts surrounding these hydrophobic triglycerides, and you have the carboxylic acid or sulfonic acid group interacting with the water. And what this allows to happen is that these collections of fats are reduced to smaller droplets. Okay, so that's sort of what the whole point of these. The bile salts are produced in the liver, they're stored in the gallbladder, and as mentioned, they are made available as food enters the small intestine. The idea here is that we want to create more surface area so that the intestinal or pancreatic lipases, these are the enzymes that break down triglycerides, have greater access to the triglycerides. So by taking a chunk of fat that's present in a piece of meat and reducing it into much smaller droplets, you allow then these lipases, these pancreatic or intestinal lipases, to access and rapidly break down the triglycerides. The issue is that triglycerides cannot be absorbed from the gut. They need to be either free fatty acids or monoacylglycerol. That is a glycerol with just one fatty acid present. So we have to remove two fatty acids through the action of the lipases to be able to get the absorption of these molecules. All right, so let's go on to the next slide. This is a diagram, and it's very complicated. I don't know if I can use, well, so let's start with number one. So bile salts emulsify, create smaller droplets of fats. These are moving now through the small intestine, and there are these intestinal or pancreatic lipases that break down the triglycerides to monoacylglycerols and free fatty acids. These can be absorbed. These can be taken through the mucosal membrane into the mucosal cells, and surprisingly, they are converted back into triglycerides. Okay, so let me restate that. We have this series of enzymes that break down the triglycerides to monoacylglycerols, free fatty acids. These are the molecules that can move across the mucosal barrier into mucosal cells, but once inside, they are reformed into triglycerides. And then they are packaged into something called a chylomicron. So if you look at the little blue circle there in the middle, a chylomicron then is this collection of triglycerides that are surrounded by phospholipids. So it has a monolayer of phospholipids to allow this very hydrophobic droplet, it's a very microscopic droplet, to enter, first of all, the lymphatic system and then into the general circulation. This chylomicron is one of several lipoprotein particles. So if you've ever had a blood test to measure your cholesterol, what you're actually measuring are levels of these lipoprotein particles. Chylomicrons, low density, very low density lipoproteins, low density lipoproteins, intermediate density lipoproteins, and high density lipoproteins. When you talk about good cholesterol, we talk about high density lipoprotein, it's HDL. When we're talking about bad cholesterol, we're talking about VLDL and LDL, low density lipoproteins. Chylomicrons are the first of these. You'll notice that there's a green blob on the chylomicron. This represents a protein, and there are actually series of proteins that are placed on these lipoprotein particles that interact with receptors or have other functions that we'll talk about in just a minute. So the circulating chylomicron then enters eventually the bloodstream. It goes through the lymphatics to the bloodstream, and you can see that red capillary there, number 6. And one of the apolipoproteins that is present on the surface of chylomicrons, this ApoC2, activates another series of lipases. Again, the problem is that the triglycerides cannot move across the endothelial layer of blood vessels into cells to be used as a source of energy. To accomplish that, we have to break these down into free fatty acids to allow them to cross into cells. And so to accomplish that, there's another series of lipases called lipoprotein lipases activated by this apolipoprotein C2, which is present on the surface of chylomicrons. This degrades the triglycerides into free fatty acids and glycerol. And as you can see in 7, these free fatty acids then move into cells where they can either be used immediately as a source of energy or they can be stored. And the primary location for fat storage is fat tissue, adipocytes. Okay, so let's go on to the next one. Okay, so this is just a single one of several bile salts. There are actually more than this, but as you can see, we have the sterol backbone, that 4-membered fused ring system. Off of the D ring, you see this extension, and this ends with a sulfonic acid group. So the ring system is hydrophobic. The sulfonic acid interacts favorably with water, so this is a detergent and is able to surround hydrophobic molecules and keep them in solution, in circulation. Okay. This is now a depiction of the chylomicron. You can see in sort of the sky blue color the phospholipid layer that surrounds the core. The core is made up heavily of triglycerides, triacylglycerols, and cholesterol esters. We won't really talk much about cholesterol esters, but they are present in these lipoprotein particles. You'll notice that there are 3 apolipoproteins. These are proteins associated with chylomicrons. Let me mention again that the C2, which is sort of this green, it's shown in green, is an activator of lipoprotein lipases, which are present on the surface of blood vessels in the circulation and allow for the triglycerides present in the chylomicron to be broken down into free fatty acids and glycerol. The B48 is a protein that is recognized by the liver. There are receptors on hepatocytes that will take up the entire chylomicron and reformulate it, repackage it, repurpose it into VLDL, very low density lipoproteins with different apolipoproteins on the surface. The C3, I don't know what its particular function is. I don't know if it has a known function at this time, but it is present on chylomicrons. You'll also notice that there's cholesterol in the phospholipid layer. Again, this has to do with its fluidity and structure, but in general, we won't really talk more about the cholesterol there. Okay, let's move along. Okay, so as we move these chylomicrons through the circulation, as the apolipoprotein C2 activates lipoprotein lipases, we will break down triglycerides, and those free fatty acids will be taken up by cells, either for use immediately as a source of energy, or the free fatty acids will be stored. Now, in adipocytes, where we store most of the fat, these free fatty acids that enter from the circulation are converted back into triglycerides. So we go to the trouble of breaking down triglycerides in the intestine to allow for absorption. We reform them into triglycerides. They circulate then as chylomicrons. We then break them down so that the free fatty acids can move into cells. And now in adipocytes, we actually reform them into triglycerides, and we place them in droplets. Okay, let's go ahead and move on to the next slide. So this is meant to be an adipocyte. Okay, so the things to the left, you can see the bloodstream, you can see a capillary there, you can see the free fatty acids moving about. But we have in the center here, in the sort of bright yellow, a lipid droplet. So this is a collection of triglycerides, but they are actually surrounded by a coating of proteins. These proteins are termed perilipins. If you look at the number 7, you can see perilipin indicated. And so we have these perilipins present on the surface of the fat droplet. This keeps it in solution. Remember, the triglycerides are very insoluble in water, and it's a little bit like vinegar and oil, okay? So, you can shake up your Italian dressing, but after a couple of minutes, the triglycerides come back out of solution, and they will stay separate. So to keep this droplet in solution, we require these perilipins, which interact both favorably with the triglycerides on the inside and favorably with the aqueous cytosol on the outside. Okay, so these represent stored fats. I have a lot of stored fats. I have a lot of these wonderful lipid droplets in my adipocytes. Now, under conditions where we experience hypoglycemia, that is, our caloric intake is low and we want to access stored fats, my body is going to produce glucagon. Okay, glucagon is going to go up. We've talked about glucagon in multiple contexts, but it also plays a role in what's called lipolysis, which is the breakdown and release of triglycerides with the release of free fatty acids into the circulation to allow them to be taken up by other cells and used as a source of energy. So this process of liberating free fatty acids and making them available is under glucagon control. Okay, so look up at number 1. We bind glucagon to a cell surface receptor. This activates that receptor. It produces a cyclic AMP. That activates our protein kinase A. Okay, we should remember protein kinase A. We saw this with glycogen. We saw this with the hepatic form of pyruvate kinase. We saw this with the bifunctional enzyme that produces the fructose 2,6-bisphosphate. This pKa has a very wide range of targets, but it is activated by glucagon. So the glucagon then activates the pKa, and the pKa is going to phosphorylate 2 groups of proteins. One is this hormone-sensitive lipase. That's pathway number 3. And the second group of proteins phosphorylated is shown by pathway 4, which is the perilipin itself. So when the perilipin is phosphorylated, it brings about a dissociation of a molecule, a protein called CGI. Okay, so the CGI now dissociates from perilipin after phosphorylation of the perilipin by pKa. The perilipins also move apart one from another. So this barrier, this coating is now disrupted. As the CGI is dissociated away from the phosphorylated perilipin, it activates the first of 3 lipases. This ATGL is the adipose triglycerol or triacylglycerol lipase. It takes triglycerides and converts them into diacylglycerol. So it takes off 1 fatty acid. That's what's shown in step 6. The hormone-sensitive lipase, once it is phosphorylated, is now catalytically active. And it will remove the second fatty acid, converting diacylglycerols into monoacylglycerols. And there is a third lipase, this monoacylglycerol lipase, MGL, which is constitutively active. It's always on, such that if I have a monoacylglycerol, it will convert it into a free fatty acid and glycerol. The fatty acids then can move out of the adipocyte into the circulation, where it will reside on a protein present in the circulation called human serum albumin, at least in humans. This albumin acts as a carrier protein. The fatty acids are have very limited solubility in water, in the blood, and so the albumin is a carrier molecule, allows the fatty acids to associate with it, and the albumin is sufficiently soluble, it's able to carry these fatty acids efficiently throughout the body. As you can see in step 10, there are fatty acid transporters in virtually all cells, allowing the fatty acid to enter. All right. Let's pause here. Are there questions?
Yeah, I'm sorry. I just wanted to verify because I was a little confused with the diagram. What are the names of the 3 lipases? I recognize ATGL. What were the other?
Okay, so there is the adipocyte or the adipose triacylglycerol lipase, which takes it from 3 fatty acids on 1 glycerol molecule to 2 fatty acids. When you have 2 fatty acids, that's called a diacylglycerol. And the hormone-sensitive lipase removes the second fatty acid, leaving us with a monoacylglycerol. And then the monoacylglycerol lipase removes the final fatty acid. It is the free fatty acids that are able to exit the cell and enter other cells. So that is the sequence of events that happen on the surface of or within the fat droplet, the lipid droplet there.
Okay, thank you.
How are the free fatty acids able to leave this droplet? Aren't they still hydrophobic? So when they
they are. They're going to leave in very low concentrations. I don't know. The location of the droplet might be closer to the capillaries than this may represent, or they may just be in very, very low concentrations as they come out and move out into the bloodstream. So.
Okay, thank you.
Yep. Okay, let's move on. Let's just skip that one. We're going to talk now about the actual breakdown of fatty acids. So this is termed beta oxidation. This happens inside the mitochondrial matrix. And we have an issue in that despite free fatty acids being very hydrophobic, they cannot diffuse across the inner membrane of the mitochondrium. And so this requires an additional series of steps called fatty acid activation. So let's go on to the next slide. So this process is happening, if you will, in the intermembrane space. The outer membrane has pores, porine channels, that allow molecules less than 5,000 Daltons to enter, pass through this membrane, and enter into the intermembrane space. But these fatty acids are unable to diffuse across the inner membrane. And so we're going to need a specialized transport system to accomplish that. So we have the fatty acid, and the initial, there is an enzyme that is going to couple the fatty acid to coenzyme A. So you can see this fatty acyl-CoA synthetase, which is going to use a copy of ATP, but the fatty acid is going to be coupled to the interior phosphate, all the way to the very inside of the ATP molecule. This leaves behind pyrophosphate, which is a very high energy molecule. It will dissociate spontaneously into two copies of inorganic phosphate. Think relief of charge repulsion. Think of delocalization, resonance stabilization for those phosphate molecules. So this very unfavorable reaction of coupling a fatty acid to coenzyme A happens efficiently because we effectively couple this to the interior phosphate of ATP. Okay. So we now have this we have this AMP attached to our fatty acid, okay? So we have, you could call this fatty acyl AMP, or as shown here, fatty acyl adenylate, or AMP. But this is not the endpoint for this enzyme. The enzyme carries out then one additional step. After coupling this to AMP, it's going to bring in a copy of coenzyme A, and it's going to displace the AMP. So AMP represents a good leaving group, and in the end of this enzyme's reaction, we're going to end with our fatty acid coupled to coenzyme A, or fatty acyl-CoA. Okay, so this represents two steps, if you will. Both of these steps occur on the same enzyme, this fatty acyl-CoA synthetase. We first produce the fatty acid coupled to AMP, then we displace the AMP with coenzyme A, and we're left with fatty acyl-CoA. Okay, so there's steps 1 and 2. The third step is going to now transfer the fatty acid to a molecule called carnitine. I'll show you it in just a second. So that would be the third step. This is not the carnitine is not a reactive molecule. So this thioester bond that we have with the fatty acyl-CoA is necessary to allow for this transfer from the CoA to carnitine. Okay, why don't we go just one slide forward? This is carnitine. You can, it's a small molecule. It has this tertiary amine, so it's always positively charged, but the fatty acid is actually coupled to the hydroxyl group. Okay, so let's go back one slide. That's why we require this very reactive fatty acyl-CoA to allow for the coupling of the fatty acid to the carnitine. Now, there is a specialized protein complex that allows for carnitine to enter or exit the mitochondrial matrix with cargo. The cargo here being that fatty acid. And it can be a long-chain fatty acid. It can be, 26 carbons long attached to this small carnitine molecule. That's okay. That will still be moved through this transporter, and that represents step 4. So step 3 is the transfer from fatty acyl-CoA to carnitine to form fatty acyl-carnitine. Then the fourth step is to move the fatty acyl carnitine inside the mitochondrial matrix. And once inside, there is this additional enzyme, which is the carnitine acyl transferase, which now moves the fatty acid back to coenzyme A, producing fatty acyl-CoA, and this now is the initial substrate for beta oxidation. So that is the fifth step, the transfer of the fatty acid from carnitine back to coenzyme A. Okay, so this is termed, this five-step process to get the fatty acid into the mitochondrial matrix is called fatty acid activation. All right.
I have a quick question.
Yes.
What's the name of the pink enzyme? The pore.
I think it's just, it's called you can call it the carnitine channel if you want. Okay. It's a translocase, so carnitine translocase. Okay.
Sounds good.
Okay. All right, so let's walk through quickly the basic steps in beta oxidation. So for a saturated fatty acid coupled to coenzyme A, there are a series of 4 enzymatic steps, and they are repeated. The last step cleaves off a 2-carbon piece, couples that shortened hydrocarbon to another copy of coenzyme A, and then the process is repeated. So we are going to clip off 2 carbons as acetyl-CoA every round of beta oxidation. Okay, so let's go to the first step here. The first of these, oops, is that the last line? Or do we have more? Okay, here we go. So step 1 of beta oxidation is an oxidation. So we start with the fatty acid coupled to coenzyme A, fatty acyl-CoA. It is acted upon by this enzyme and produces a trans double bond, okay? So we talk about trans fats being bad. That is true, but not because of their, well, those are problematic in the circulation and not inside the mitochondrial matrix. Here we actually form a trans double bond between carbons 2 and 3 of the fatty acid, and by this enzyme. We also produce a copy of FADH2. Okay. All right. The fatty acyl-CoA dehydrogenase is actually embedded in part in the inner membrane. And this actually, you can see the orange or reddish orange blocks. This represents a series of FAD molecules that feed the electrons through finally a iron sulfur cluster directly to coenzyme Q. So the FADH2 molecule that is produced releases those molecules to other FADs in the electron transporting flavoprotein, ETF, onto iron sulfur cluster and onto Q. So that's step 1. Step 2 is a hydratase. This is a lyase reaction in that we're adding water to a double bond and producing a hydroxy species. So we add this and we end up with this beta hydroxy acyl-CoA molecule. So the name of this enzyme is ene, so the double bond is an ene-OEL CoA hydratase. And it produces the beta hydroxy acyl-CoA. This third step is another oxidation. And that hydroxyl group is going to be oxidized to a ketone. So we go from the beta hydroxy to a beta keto acyl-CoA. And this beta-hydroxyacyl-CoA dehydrogenase is also going to produce a copy of NADH, which will feed into the electron transport chain. In the final step, there is an enzyme. We will call it thiolase. You could learn the longer name, but we'll just call it thiolase. So we have this beta-keto-acyl-CoA, and this enzyme brings in a copy of CoA and couples that to the carbonyl of the beta-keto group. So it truncates the fatty acid hydrocarbon chain at that keto group and leaves us with this 2-carbon piece, which is acetyl, an acetate coupled to coenzyme A. And we've coupled the remnant, this keto group now, to a thiol. So we actually have taken the keto and converted it to a carboxylate, producing a thioester with coenzyme A. And we release acetyl-CoA, which feeds into the citric acid cycle. We have then shortened our fatty acid by 2 carbons. But we have a fatty acyl-CoA, and it can feed back into the beta oxidation pathway and can be acted upon by the fatty acyl-CoA dehydrogenase. Okay. Sorry for the scrambling, everyone, but we'll stop here. I will. Let's see. We have a time for a couple questions. If not, I'll continue my story. It's a little odd doing this at my desk, but we'll do the best. Anyway, so during my second year of grad school, I had these 2 oral exams. The first one was on my research, and it was very focused. The second was this general oral that could be on anything at all. So I took my first oral, which was related to my research, and I passed it. That was not a surprise. Most of the concern there is whether the experiments proposed seem to be adequate, whether the project seemed to be sufficiently focused that it wouldn't go on forever and ever, whether there are alternate plans, research plans for doing it. The second oral now, though, was very different. It was anything goes. Anything that my research committee could think of to ask, they could ask, and I was supposed to know the answers. On my committee it was a 3-person committee. I had my research advisor. I had one other faculty member who was in the same area, and I had the third one was a person in our department, a faculty member who had a reputation. In fact, his nickname was Black Jack, he had a reputation for being difficult, not only as part of a graduate student's oral committee, but just in general as a person. He seemed to be a difficult person. I think I will stop here, set up the thing, so we're pretty much out of time and I don't want to run over. I apologize for all of the confusion at the beginning. I hope this got recorded, and I will try to get this posted as quickly as possible. I will be doing my office hours in just maybe 10, 15 minutes. I will open that up for people, and we'll go from there. Thank you very much. Thank you for your patience. I may be back in the MARB next time, or I may not, so we'll see. So.